Okay. So I'm Emily.
And I'm Martha.
And our article is called Protective Effects of Dietary Supplementation with a Combination of Nutrients in a Transgenic Mouse Model of Alzheimer's Disease. So basically our study looked at the effects of diet on Alzheimer's disease.
So first of all, we're going to talk about what is Alzheimer's disease. It is the most common form of dementia, and the causes of it is that there are amyloid plaque buildup on the outside of the neurons, and there's neurofibrillary tangles, so basically the neurons cannot signal each other anymore, so connections begin to be lost. And some of the symptoms is that there's memory loss, impaired cognitive functioning, a decline in ability to perform day-to-day activities, and changes in personality and behavior. So as you can see, this could be a big problem in people. So first in this paper, what they talked about, that there's no treatment for Alzheimer's disorder, so right now they're all focusing on ways of preventing it, and in this paper they're looking at nutrients specifically. Previous studies looked at nutrients that help prevent Alzheimer's disease individually, and these are a list of nutrients that they have seen before that they help, and they're like DHA, EPA, PS, antioxidants, macular carotenoids, and date palm fruits. Go to the next slide. So what do these nutrients do in general? So they reduce amyloid beta accumulation, which I've mentioned, but it's one of the things that causes Alzheimer's disease. And there's neural repair and protection, modulation of intracellular signaling and metabolic pathways, improve cognitive functioning through regulation of receptors, enzymes, ion channels, and signaling molecules. And then they reduce this ROS-induced damage, stabilize the memories of the neurons, improve visual functioning, oxidative stress reduction, and improved memory. So the aim of this study specifically, as they said, is to investigate the effects of dietary supplementation with these nutrients that are listed there, and on the cognitive function, oxidative stress, and amyloid precursor protein, presenilin, and double transgenic mice. And the nutrients that they could lead to against the cognitive defects and cellular damage in the onset of the development of Alzheimer's disease. So basically they wanted to see how a combination of all these nutrients to be in their meal at once will help instead of individually.
Okay, and so they used a lot of materials to be able to do this experiment. And so they used the Water-Morris maze, they used the platform, they used a shuttle box device, and they used 72 double transgenic mice, as well as 24 wild-type littermates that served as a control. And so in regards to the diet, they were randomly divided into 3 groups of 24 animals, and there were an equal number of males and females per group. And so there's a model group that just ate normal chow. There's a high treatment group, and that was, they received that dosage of those nutrients at the highest dosage that is recommended for intake for humans. And then there's a low treatment group, and that was just half of the high dosage. And then there's also the negative control, which was just those wild type mice fed normal chow. And so in regards to the Morris water maze, there was 12 mice from each group that participated in this. And this happened at both 3 and 7 months after treatment began. And so the Morris water maze is used to measure the spatial learning and memory of the mice. And so there are 4 quadrants that are surrounded by black curtains, and they need to be able to locate this platform. And so they do training to locate this platform for 5 days, and then after that they have this one test day where they remove the platform and they see if the mice go to see if the platform is still there, essentially. And then they also use the shutterbox escape and avoidance task. And so this consists of 2 equal-sized compartments that's connected by an opening. And what will happen is that a light will shine for 20 seconds, and then following that there will be a 10-second shock stimulus that goes through the floor. And so a round is complete when the mouse crosses from one side to the next. And so crossing over is considered either avoidance or escape. It's avoidance if the mouse crosses over when the light shines, and it's escape if the mouse crosses over when that shock runs through the floor. And the mice participated in one single session of about 40 trials, so 40 times going back and forth. As far as biochemical parameters, they took 12 mice per group in the behavioral studies after the 3 and the 7 months of treatment, and the mice had fasted and were anesthetized and blood drawn for biochemical analyses. And then the brain was removed from 6 animals per group, and the left side of the brain was ground and homogenized and centrifuged to be able to measure some more of those biochemical parameters and levels. And then they also took 6 different mice per group and removed and fixed the brains for histological and immunohistochemical studies.
So first of all, there was no significant difference on the daily food intake, which is a good kind of control to remove confounding variables. So basically, these are all the 4 different types of groups that they looked at, and they all weighed the same, and they all ate the same amount. So that's just really good to notice in the research that that wasn't something that could have contributed. So then for the behavioral test, for the water maze test, for the intervention at 3 months and at 7 months, the high treatment group and the negative control group, the high treatment group and the negative control group for both times, they were lower at figuring out on time as to where the little platform is at, compared to the other 2 groups that had the low food nutrient intake and the model group, so that they took longer to figure out where was their safe place. And then on the 6th day, as Emily had mentioned, when they removed the platform, it showed the same information, that the high group and the negative control group, they both found the area where the platform should have been faster than the other 2. And then for more behavioral tests, for the shutter box escape avoidance test. So escape failures percentage across groups at 3 and 7 months. As you can see, that there's a higher escape failure for the groups in the model group compared to the other 3. So then the ones who were in the negative control and didn't receive any form of the nutrients all had fewer escape failures. And then for the active avoidance, as you can see, the MG group, so the model group, they were the ones who struggled the most with the active avoidance at 3 months, but then at 7 months, both the model group and the low treatment group both had the lowest amounts of active avoidance as compared to the other 3. Okay. And then for this are results for the ACH and the total cholinesterase levels in the serum. So in the ACH in the serum between groups according to 3 and 7 months of intervention, as you can see, it was high for the high treatment group and the negative control group at 7 months and at 3 months for both of them. And then here we're looking at TCHE in serum between groups according to those 2 months, and it was higher for both 3 and 7 months for the model group. Then for ACH and the TCH levels in the brain now, not in the serum. So for in the brain, it basically showed around the same thing, that it was lower for the model treatment groups for both 3 and 7 months compared to the low treatment, the high treatment, and the negative control. And then the same thing here for total cholinesterase in the brain, that it was the highest for the model group compared to the other 3. Okay. And then for the results for indicator of oxidative stress. So there's a total, for the total protein in the brain, it was the highest for the model group, especially at 7 months of intervention. And then for the GSH-PX in the brain, it was the lowest in the MG group, especially like at the 7 months. I mean, both 3 and 7 are lower compared to the all 3. For MGA in the brain, again, you can see that for the model group, it was the highest. So if they had any form of nutrients, or they're in the negative control group, they had lower amounts of MGA in the brain. And then for MGA in the serum, it shows basically the same thing, except that for the low treatment group and the negative control group, it's a little bit higher in the 7-month area, but not as high as the model group. For the SOD in the brain, the model group was the lowest compared to the other 3, but as you can see, like the low treatment group and the negative control group had higher SOD in the brain than the high treatment group of the nutrients. And then for the serum, it shows the same thing, that the model group is the lowest compared to the other 3, and they show that like the amount of nutrients, it stays around the same for the 7 months, but it's a little bit lower for those 3 groups. And then for the last of the results is that pathological changes in the brain. So for the H&E staining, at 9 months, fewer neurons and pycnotic neural nuclei in cerebral cortex and hippocampus of transgenic mice, but not in the wild type. So the ones that received the treatment had the lower amounts of these types of neurons. And for immunohistochemistry, they showed that there was an increase in the number volume of amyloid plaques proportional to the age in the mice. So basically, that yes, there were increasing amounts of those causes for the Alzheimer's disease. And then at 7 months, there were fewer plaques in the cortex and hippocampus of the low treatment group and the high treatment group compared to the model group. So it's saying that even like a half of the dosage of the nutrients or the total amount of nutrients both helped remove some of these plaques in the cortex.
Great, so Martha just went over a ton of results, so we're going to see how this actually applies to the hypothesis and what we wanted to get at from the result of this study. So basically, this study suggested that their hypothesis that cognitive functioning and biochemical and histological indicators improved after consuming these nutrients for 7 months. And this is great. This is what we wanted because it tells us that the memory and cognitive functioning declines that we typically see with Alzheimer's disease were in fact improved through these dietary interventions and the addition of the nutrients. And so this suggests that diet can be used to delay and even prevent Alzheimer's disease. And so we know that oxidative stress increases decay and can be exacerbated in Alzheimer's disease because it's involved in the pathogenesis and progression of Alzheimer's. This is due to the attack of lipids and proteins and sugars and nucleic acids by those free radicals. And so the byproducts as a result of these attacks serve as markers of damage by oxidative stress. And in this study, these markers were a lot less apparent in those diet groups. We also saw that a decrease in ACH contributes to memory dysfunction in Alzheimer's, and those nutrient groups, in fact, had higher levels of ACH, which improves memory functioning. And it also appears that these nutrients have protective effects, which is really important because these protective effects promote neural repair and it inhibits formation of those amyloid plaques, and those amyloid plaques result in those tangles that cause the neuronal death and dementia, and so that's very important. Overall, the future research needs to be done to more clearly define the beneficial impacts of diet and certain nutrients on Alzheimer's, because Alzheimer's can be such a pervasive and exhausting disease, and treatments aren't always that great or cost effective, and so learning more about the effects of diet on this disease can really improve how we look at Alzheimer's and treat it across the world. Thank you.